Hello friends, welcome to some biology class. Today we shall discuss about different types of enzyme inhibition. Enzymes are the biocatalysts. They increase the rate of biochemical reaction by lowering the activation energy. Any substance that decreases the velocity of enzyme catalyzed reaction is known as inhibitor, and the process is called as inhibition. The enzyme inhibition can be broadly divided into two different categories. They are reversible inhibition and irreversible inhibition. The reversible inhibition can be competitive, non-competitive or uncompetitive. In this type of enzyme inhibition, the inhibitor form weak and non-covalent bond with the enzyme. As a result, the inhibitor is easily dissociated. What is competitive inhibition? In this type of inhibition, the inhibitor closely resembles the substrate. That means they are structurally related. As a result, the inhibitor competes with the substrate for binding at the active site of the enzyme. That means the competitive inhibition occurs at the active site, and the degree of competitive inhibition is proportional to the relative concentration of the substrate and the inhibitor. This type of inhibition can also be overcome by increasing the substrate concentration while keeping the concentration of inhibitor at a constant level. In competitive inhibition, the inhibitor can bind only with the free enzyme to form enzyme inhibitor complex, but the inhibitor cannot bind with enzyme substrate complex. In this type of inhibition, the maximum reaction velocity remains unchanged, but the Km value increase. What is Km? It is the Michaelis-Menten constant. It is equal to the substrate concentration at which the reaction rate is half maximal. Second type of reversible inhibition is non-competitive inhibition. In this type of inhibition, the inhibitor binds at a site other than the active site of the enzyme. The specific site at which the non-competitive inhibitor binds is known as allosteric site. Because of this, the inhibitor can bind with enzyme to form enzyme inhibitor complex, and the inhibitor can also bind with enzyme substrate complex to form enzyme substrate inhibitor complex. The formation of enzyme substrate inhibitor complex is possible because the substrate and the inhibitor bind at different sites. This is about non-competitive inhibition. Third type of reversible inhibition is uncompetitive inhibition. In this type of enzyme inhibition, the inhibitor also binds at the allosteric site, but this type of inhibitor can only bind with the enzyme substrate complex to form enzyme substrate inhibitor complex. Second type of enzyme inhibition is irreversible inhibition. In this type of inhibition, the enzyme is permanently inactivated and the inhibitor tightly binds with the enzyme. As a result, it is slowly dissociated from the enzyme. Such type of inhibitors form both covalent or non-covalent bonds with the enzyme. And in case of irreversible inhibition, a specific functional group of the enzyme that is crucial for its activity is permanently inactivated. So friends, this is all about different types of enzyme inhibition. For more updates, please subscribe my YouTube channel, Sam Biology Class, and also please share your comments. Thank you very much.